Hi everyone, in today's lecture we're going to be talking about genetics, so let's first get into what is genetics. Genetics is the scientific study of heredity, genetic variations, and genes in living organisms. So first let's break down some key terms. So scientific study of heredity. Heredity is how we pass down genetic factors, so our traits. So biological parents pass down traits to their offspring, and then also, we're looking at genes in living organisms, so these are different units of heredity, and they're basically the building blocks for our body, and they act as the instructions on how to make up our body parts, and they're segmented into DNA. And on the next few slides, we'll get more into genes. We also look at genetic variations when we're studying genetics, and so this looks at the diversity of our genes and how we are different within different species. And so this is looking at our different hair and eye color. And so I included a couple videos that have basic information on genetics and heredity. So I would recommend that if you need more information on those two key pieces of genetics, I would recommend checking those out. Also, we see that we have the Human Genome Project, which is something that was essentially completed in 2001, but there is still analysis that's ongoing, and this involves basically an international effort to kind of map out the complete human genetic code, and it studies all of our DNA and looks at genomes, and basically the genomes are all of our genetic material within ourselves as living organisms. And so this is something that we do to look at each individual member of a species. All right, so on this slide, you'll see that I've included an image of a DNA structure, and I wanted to show this first because it kind of helps us to visualize what we're talking about. So basically, the way we can think of it is that we each have a cell, and within the nucleus of the cell, which is the powerhouse area of the cell, we see that we have chromosomes. And on the next few slides, we'll talk about the chromosomes. And within each chromosome, we have DNA, and then parts of our DNA are called genes, and so each gene then has different protein bases that they are pairing together in order to make up us as individuals. And so this is a visual image, and I would recommend referring back to this just to kind of map out where each piece of the DNA structure falls. And then on the next slides, we're going to get into chromosomes, DNA, we're going to get into genes, and we're going to get into the nucleotide base pairs, also known as our proteins. Alrighty, so remember, when we're thinking about genes, we're thinking of a unit of heredity, and so these are the building blocks for our bodies, and they're segments of DNA. So in order to understand genes, we need to understand our DNA, or our deoxyribonucleic acid. And so that's a mouthful, so we just typically say DNA. And so basically, all living organisms, they're composed of cells. Remember the image that we were looking at on the previous slide. And each one of those cells has chromosomes, and within those chromosomes, there are instructions called DNA. And so basically, our DNA is a molecular structure that contains chemical instructions for cells to manufacture different kinds of proteins. And so we see that methylation is this process that helps us to transcribe, connect, and alter our genes. And so there's this turning off of gene expression, and it's basically our bodies telling us whether or not this certain kind of gene is needed for us as human beings. And so when we're looking at our DNA, I like to kind of consider DNA as a book. And so it's the unique code used to write our cells. And again, this is something that promotes and sustains life, and it's formed within chromosomes. So chromosomes are going to be what we discuss on the next page, and then on the next slide, excuse me. And if you were to look at the previous slide, it's basically that X-looking shape. And so DNA is a big, long structure within our chromosomes. And so genes are the segments of DNA. So if we have our DNA, we have to think about our genes as the short pieces of DNA. So remember I said, think about DNA kind of like a book. Well, if we have a book, what's inside the book? pages. So I like to think of the genes as the pages. This is how we organize all of that information that is written in code is organized within those pages. And so genes are short pieces of DNA, and they have specific sequences of nucleotides. And on the next slide, you'll see that we're going to talk about those nucleotides and their bases that are found within the DNA molecule. And so DNA, again, has those two strands, and it's called a helix formation. And they carry out the instructions. And so remember on the first slide, when we looked at genetics, we're looking at heredity, genetic variations, and genes. And so something about genes that we want to consider is not just each segment, but the entire set of our genes. And so that's when the genome comes into place. So that's basically like thinking about all the books put together. If you have one book that shows your DNA and the genes within those pages, think about a genome as the entire book series, and that's a complete set of all your genes. And so on the next slide, we'll get more into the proteins and the chromosomes. So remember, we have our cell, then our chromosomes, our DNA, and then our genes. Alrighty, so remember, when we're thinking about human beings and we're thinking about the molecular structure of our DNA, it's broken off based on the chromosomes. And so chromosomes are long chain sections, and they're thread-like structures made up of proteins, and these are also made up of single molecules of DNA, which we just previously talked about, how our DNA is different instructions for how our genetic code is made up. And the chromosomes carry genetic information from cell to cell. So if we're thinking about one cell within our body, we have a set of chromosomes in that cell, and DNA consists of 46 chromosomes arranged into 23 pairs. And we have other 22 pairs are autosomes, and those are inherited equally by both males and females. And 44 of those autosomes and 2 of them are sex chromosomes. And so the sex chromosomes that individuals have determine whether or not you're going to be male or female. And so these are X and Y chromosomes. And so females are XX and males are X and Y. And during conception, male and females contribute to the sex chromosomes. And so the male is actually the one that determines the sex of the baby. So you'll see I've put a little image here, and I've also included a video that kind of talks about the process of how chromosomes are actually formed and how it's a little bit more complicated than just XX and XY. And basically, we're seeing that the sex of the offspring depends on whether or not the father's sperm is X or Y. And then this fertilizes the ovum or the egg. And so again, the father is the one who determines whether or not you're going to be born with XX or XY chromosomes. And we also see that the sex is determined around 4 to 6 weeks of gestation is when we're able to kind of see that. And we also see that within our cells, we have proteins. And so proteins are different strings of amino acids with adenine, thymine, cytosine, and guanine. And so those are within the genes, and these are different nucleotide bases, and they replicate and transcribe information. And so these are most similar in humans, but we see that our DNA code is what makes us different. And so most of the time our proteins are going to be exactly the same because we replicate and transcribe information in our bodies similarly. However, our DNA is actually going to be slightly different. And we'll also see that there's different bases that we have, and they're arranged in multiple ways. And so although we're always going to have AT and TA paired together, as well as CG and GC paired together, there could be more than 3 billion base pairs within one human being. And so we see that there are many different variations of how our proteins replicate and transcribe information. And so I've included another little video here that kind of just helps you to remember these proteins and how they're paired together and what they mean. Alrighty, so as you can see here, the image on this slide shows some different chromosomes. And so again, there are those X-looking shapes in our DNA makeup. And so remember, when we're looking at genes, we have our genotypes. And so I mentioned that previously we could see on the slide we had genotype and phenotype. And so genotype is again just our genetic makeup, and we also see that we have different types of genes. And so we see we have homozygous and heterozygous genes, and basically that just indicates for homozygous it's just two gene pairs of one that are exactly the same for their code, and most genes are paired like this. And we see though that we sometimes have heterozygous genes, and so this is when two genes of one pair differ in some way. And typically this is when we have an allele that has a different base pair than another member of the group within that. And so this is when we're thinking about dominant and recessive genes. Sometimes we have pairs that are put together differently. And so this can then determine whether or not our phenotype is going to look different from one another. So even though we may have a similar genetic makeup, if we're thinking about maybe siblings, for example, we may have different ways that those kinds of genes are then produced and processed. And this could be due to the different kinds of codings within our genotype. All right, so let's talk about gametes and zygotes. So gametes are our reproductive cells. So this is in any organism, there are reproductive cells. And so that looks like our sperm and our ovum. And so we think about those different kinds of reproductive cells engaging in the process of mitosis. And so this is when we have cell duplication to create a new body of cells. And you'll notice that I also have the term meiosis. So there's a process of meiosis and a process of mitosis. And so both of these occur in organisms, and they help produce new cells. And they have similar steps. The only difference really is that we see with meiosis, it's the process of two gamete cells, whereas mitosis is just the process and duplication of one cell. And so a zygote is what we think of when a new baby is being formed. And so we see that zygotes take two gametes, which are going to be our sperm and our ovum, or our eggs, and those are two cells that are put together and create a new cell. And so we see that hours after conception, between sperm and ovum, that we have millions and millions, trillions and trillions of cells that are being formed. And at birth, a zygote becomes 10 trillion cells. And so basically we see that we are creating, with 23 chromosomes from each parent, a new cell. And so we see that each cell carries out its own complete genetic instruction copy. And so you'll notice that when we get into discussion later, that there may be some variations in how this formation comes to be. So depending on that cell division within the meiosis process, when a sperm and an ovum are being combined together, we may see that cell division occurs in 2s, 4s, and in 8s, and then this can create different kinds of individuals. So this is where we start to see twins and quadruplets or octuplets. So we might have extensive cell division within the process of forming a new organism, and this creates a variation in what happens during the duplication of human beings. And so I've included a song here that kind of helps you to understand the process of meiosis, just for fun. And then on the next few slides, we'll talk a little bit more about siblings versus twins, and we'll start to get into some specific interactions between genes. So once we have the formation of a zygote, it begins the process of new cells being formed, and then we see that those cells being formed then create the beginnings of individual genes in human beings with different kinds of alleles. So those are those dominant and recessive traits. Again, those are formed from our parents. So we get different kinds of heredity or traits from our parents, but we got to start out with mixing those genes together to create new life. When thinking about conceiving and sustaining a pregnancy, we often have to consider different techniques that might be used, especially for our infertile couples. And so something that we use is assisted reproductive technology, and this is a way to support men and women who have fertility problems. And so 1/3 of women and 1/3 of men suffer with infertility, and then 1/3 is due to unknown causes. And we do see that there is fertility that decreases with age, and there's a faster decline for women. And infertility is something that happens when there's a failure to conceive after 1 year of trying. And we see that there are different types of fertility treatments that can be done on eggs or embryos and how they're manipulated. And so sometimes there's the use of a donor egg or donor sperm or even frozen embryos. And this is created after fertilization of a sperm and an egg. And we also see that sometimes this involves surrogacy. So if there is difficulty in sustaining a pregnancy or infertility altogether, we may see that a surrogate is used, so the eggs and sperm are put into another person's body in order to carry out the pregnancy, but this could be the egg and sperm from the mother and father, and it's just put into basically a housing unit for the pregnancy. So the bun in the oven is going to be in someone else's body. However, the ingredients that were made to make that bun come from mom and dad. And mom and dad do not have to be the surrogate. We also see the most common form of reproductive technology is going to be in vitro fertilization. And so you can think of this as something that takes place outside of a woman's body. And this happens within the lab. So we take the sperm and we take the egg and we put them together kind of in a petri dish, and then when you mix them up, this is when we can create the zygote. And so this is done within the lab because there is some form of infertility within either the male or female, and so they're not able to produce this on their own within inside a woman's body. And so then the zygote is implanted, it's fertilized, and it's implanted into a woman's uterus so that the baby is then ready to grow. And there are some complications that can happen with this. So we may see increased birth defects and later illnesses. And we also see that almost half of babies who are created through IVF do have low birth weight. And we also see that there are more instances of twins or triplets, which we'll talk about twins on the next slide. All right, so on this slide, we're talking just a little bit about twins. So twins are just a kind of variation of siblings that happen within one pregnancy. So typically when we're thinking about siblings, we think about one pregnancy, one baby is born, and then within the next time period of the pregnancy, another baby is born. Whereas with twins, they are growing within the body simultaneously. So monozygote twins are identical twins, and they originate from one zygote. And so this one zygote splits very early in development. And we do see that if the split of the zygote is incomplete, this could result in twins being conjoined. So again, they are forming within one egg. And so identical twins, there is a split of that one egg, and so there could be complications during that split, and then this results in twins being conjoined. We also see that there is the exact same genetic code makeup. So the genotype of identical twins is the exact same. However, there could be some slight variations in the phenotype due to environmental influences. So once we are within the environment, there may be changes within our genes and how they are then represented. And so I have included identical twins separated at birth teach us about genetics. So interesting video to kind of talk about how variations within the environment can strongly influence twins, even though they have the exact same genetic makeup. This may be expressed differently depending on the kind of care that they're provided with parents, the kind of school systems that they're in, all of those different environmental factors, stress, trauma, all of that impacts a human. We also see that fraternal twins are dizygotic twins. They result from not one, but two eggs. So identical twins are going to have one egg that splits, and fraternal twins are going to have two eggs that are just fertilized at the same time. And so we see that just like the twins in a fraternal split have two eggs so do their genes. They split in two. So fraternal twins are more commonly genetically made up, just like regular siblings. So we see that genes are split in half. This is why we might see some variations in phenotype. So fraternal twins often will have similar characteristics in the way that they look and present themselves, but they're not going to have identical characteristics necessarily in how they are presented. We also see that fraternal or dizygotic twin dynamics are much more common, twice as common as a identical pairing. All right, so on this slide, we're diving a little bit more into the phenotype. So remember, we have our genotypes, which are the genetic expressions of different kinds of genes that we have, and the phenotype are the observable characteristics of an organism. So this is our appearance, personality, other traits that are passed down to us from our parents. And we see that we have dominant and recessive alleles. So these are just different factors of our genes. And you may be familiar with or have practiced a little Punnett square before. And so this is something that we commonly do to identify dominant and recessive genes. So again, this could be eye color, this could be hair color, this could even be, the height that we are. And so we're looking at different alleles to distinguish individuals' phenotype. And so we see that there are different variations, and that there are some that could be life-threatening depending on what kind of genes we're looking at. So this could be cancerous traits or things like that. And we see that many genes are identical for every human being. So we have many individuals who have blue eyes, many individuals who have brown eyes, same with, the kind of texture hair that we might have. There are similarities within the patterns of our genetic makeup, but because we have so many different kinds of individuals out in the world and different people who end up interacting and then making babies, we have a lot of variations within those genetic codes. And so over time, we have more variations within what that looks like and the kinds of codes that we can have. And we also see that our adaptation within our environment could potentially change the expression of these genes. So remember, we kind of talked about epigenetics. I would recommend going back and looking at that video to just kind of refresh your memory if you need to on how the environment can impact our genetic variations. And we also see, again, that dominant and recessive heritability. And looking at dominant genes are more influential than recessive genes. So dominant means that it's going to be presented more strongly, and recessive means that it's not going to be presented as strongly or not at all. So it could be a non-additive within the variation of our genes. And we also see that the dominant gene can completely take over the phenotype. So if we were looking at blue eyes versus brown eyes, many brown-eyed people often carry a recessive gene for blue eyes, but it's not expressed as commonly. And fun fact, we also see that for blue-eyed parents, that there is the blue eye and brown eye gene. So brown eyes are dominant and blue eyes are recessive, and this could actually result in hazel eye combination for our phenotype. Last but not least, let's talk about some chromosomal and genetic problems that may occur. And so on your text, it shows table 3.1. It has a little bit more information on common genetic diseases and conditions. And the reason why it's important for us to discuss this is that sometimes we may have differences, and this doesn't always mean that there is a deficit underlying. However, we do need to know the complexities of our genetic makeup in order to provide the best support and know the different effects and kind of what is the outcome of differences in our genetic makeups. And so there are dominant disorders, and so we see that about 7,000 known single gene disorders are dominant, and this is evident within the phenotype. So we often see that there is a change in the way that a person looks or the way that their traits are visibly based on a chromosomal and genetic variation or difference. And we also see that there are some recessive disorders. And so we're going to talk about Fragile X syndrome, sickle cell trait, Down syndrome, and cystic fibrosis. And so we see with our Fragile X syndrome, so FXS. This is caused by more than 200 repetitions of one triplet on one gene. And so this is most commonly a form of an intellectual disability that is inherited. And so this could produce developmental delays such as walking or talking. This also is connected directly to learning disabilities. We also see that social interactions may vary and behaviors such as eye contact, attention, or even outbursts in our behavior. And we also see that females have more mild symptoms within Fragile X syndrome than males do. Now, if we're looking at sickle cell trait anemia, as well as if we're looking at cystic fibrosis, both of those are recessive disorders, and there are several types of these that are sex-linked traits exactly. So let's talk a little bit about sickle cell trait anemia first, or SCA. So this is actually an abnormality in the oxygen-carrying protein, hemoglobin. This is found in our red blood cells. And so the red blood cells are different and they often cluster together. And so someone with SCA will have blockages in these blood vessels. And so this actually decreases healthy oxygen flow. And so this chromosomal change happens as a mutation of the gene, and it does actually offer some protection against malaria. So having sickle cell trait, because you're not as likely to get sick due to having red blood cells that make it harder for you to have infectious malaria growing, because there's blockages, and so you're not as likely to get sick from malaria. So I did include I did include a link for you here to kind of learn a little bit more about sickle cell trait and how it can protect against malaria. And we do see that individuals who are African American are more common to carry this kind of abnormality in their gene expression. And for Down syndrome, we do see that this is also called trisomy 21 because a person has 3 copies of the chromosome 21 instead of having 2. So typically we have 2 chromosome copies, but somebody who has Down syndrome will have actually 3 copies of that. And so this is basically called Down syndrome, and you can have extra chromosomes, and the missing chromosome is called a monosomy, so mono meaning 1. And we see that there are specific variations within the phenotype. Again, this kind of variation in gene expression, there are common physical differences. So a person with Down syndrome will often have almond-shaped eyes. They may have a shorter neck or smaller ears. Excuse me. We also might see some typical behaviors related to Down syndrome in children. There may be more likelihood that they will wander off. It might be a little bit more difficult for them to follow instructions, as well as engage in impulsivity and aggression. And last but not least, with cystic fibrosis, this mostly affects the lungs, but it also can affect the pancreas, the liver, and intestines. And we see that airways are blocked. We have more sticky mucus, and they're damaged due to being kind of stuck with all of that mucus, and this happens, and then germs are more likely to be trapped in there, and this can lead to a higher risk of infection. And we see that then we're not able to absorb as much nutrients, and we see that babies are often checked for cystic fibrosis as newborns, and it's more common in Northern Europeans, and we see that carriers may be more protected from cholera, which is a bacterial disease, and so this is often common when eating foods that have contamination, and it often affects the lungs, so it actually helps to clear the airway. So having that mucus build up and break down can kind of protect you from common contamination within the bacterial elements of the food. So we see that both cystic fibrosis and sickle cell trait do have some benefits related to other diseases out there. However, these are common chromosomal and genetic problems. Like I said, see table 3.2 for more common genetic diseases and conditions. And this is all I have for you for today's lecture. So enjoy, and I will talk to you next time. Bye, everyone.